Okay, let's go ahead and start. What we're going to do today is some actual hands-on experiments. I think if you're zooming, you should be able to see the slide. It says Unit 1, Week 2, membrane transport and permeability. So we're going to go back and pick up the other tissues, and we're going to give you some time to make some cheek cells to show that you really are alive and have real cells. But while that is, while we're going to be doing that, I have another experiment running over here. I want to talk about that first, and then we will get everything wrapped up. Okay, so let me bring this up to full screen. But membrane transport and permeability, what are we talking about here? What it is how do cells get stuff inside or outside the cells? And okay, nightmare. How do things get into a cell and outside of and out of a cell? You know, cells are pretty well contained little structures, and you know they got to have some way to get things moving. Cells are isolated units. They're the smallest living unit in our bodies. The cells are alive. The cells are surrounded by salt water. We call it interstitial fluid. See, the interstitial fluid comes from our blood. Our red cells and our white cells float in our arteries and veins in a fluid called plasma. Plasma is 0.9% sodium chloride, salt water. Well, the capillaries, the arteries, and the veins are all leaky, and the fluid leaks out. And when it leaks out of the vessels, which it does all the time, it ends up surrounding the cells, and we call it interstitial fluid. The interstitial fluid is salty, just like the, thank you. The interstitial fluid is salty just like the plasma was, because a few minutes earlier, it was plasma. Before it became interstitial fluid, it was plasma. And bless you. So, but it leaked out. Now, contained in that interstitial fluid, we have salt, sodium chloride, we have sugars, we have fats, we have hormones, we have proteins, we have all sorts of stuff in the interstitial fluid floating around that surrounds the cells. Now, that fluid doesn't necessarily go into the cells, but they're bathed in it, they're covered with it all over the place. And the reason that stuff doesn't go into the cells is because the cells are also filled with water. The same salt water that surrounds the cell is the same type of salt water inside the cell. Now, a cell will allow things to come into it or go out of it as it's needed. And it's free. It's a free ride. It's totally passive. Movement across the membrane of a cell from the interstitial fluid is totally free, because cells are cheap. I'm sorry, cells are frugal. Cells don't like to spend energy. So if you're a cell and you need more salt, for example, and there's salt outside, you'll find a way to get it in. If you have too much water and you get rid of it, you'll find a way to get rid of it. It doesn't cost you anything. The process that we use is called diffusion. Diffusion is a pretty simple term here. It means diffusion is simply the movement of a substance from a high concentration to a low concentration. That's all it is. You know, we move from a high concentration to a low concentration. It can be in water, it can be in the air, it can be in a gel. It doesn't matter. A molecule will move passively from its high concentration to its low concentration because for some reason they want to reach equilibrium. If I dumped a bottle of some sort of chemical, there's a whole bunch of chemicals used in food science. One of them is artificial banana flavoring. And it's also used when you test, for respirator packs like the fire department has, to make sure that there's no leakage. So they'll pour a little of this on the floor around where they're standing with their mask on. And do you smell bananas? It's like artificial banana oil. It's not unpleasant, but it's very penetrating. If I were to dump a little of that artificial banana oil on the floor over here, pretty soon everybody in the room would smell bananas. And because it would start out strong over here and gradually spread as it's carried by the air molecules. That's diffusion, from a high concentration to a low concentration. So it's passive. The only time a cell doesn't go with passive is when it has to use a special carrier protein that requires energy. So we're not talking about that right now. So membrane transport, passive transport. We see in the slide here, diffusion through the lipid bilayer. That's a cell membrane. Well, diffusion is very passive, very simple. We simply go from the high concentration to the low concentration. Gases do this all the time. We breathe in outside air, okay? And outside air is 20% oxygen and 80% nitrogen. In our blood, outside our lungs, the oxygen level is about 16%. Now, we realize that we have 20% oxygen in our lungs and 16% oxygen in our blood. We're going to diffuse from the lungs into the blood. It's passive because we have more oxygen on one side of a membrane than on the other side, so it just slides downhill. We always have more oxygen in the air than we have in our lungs, so our lungs always send oxygen to the blood. So it's a free ride, and our bodies love it. Yeah. So in the cells, we have diffusion. Most things diffuse. Oxygen diffuses from outside the body ultimately into the cells. Carbon dioxide diffuses out of the cells and into the lungs because there's always more carbon dioxide in blood than there is in the air. We want to get rid of the carbon dioxide. We don't need to have it in our bodies, so we get rid of it. The cells generate carbon dioxide. It goes into our blood. The concentration in our blood is higher than what's in the outside air, and so we go from the high concentration to the low concentration. At the same time oxygen's coming in, carbon dioxide's going out. Okay, a couple terms. You'll hear more about this in lecture. A solution is a mixture of at least two parts, two components. It consists of a solvent and a solute. Now, water is universal solvent. We dissolve things in water. If you make iced tea, you dump your sugar into the hot water, right? Everybody knows how to make tea. You dump. The sugar is the solute. The water is the solvent. The water dissolves the sugar. If you add salt to water for cooking, what happens to the salt? It dissolves, right? The solvent is what we call the dissolving medium. It's always in our bodies, it's always water. The solutes are the things that are dissolved. So a solution is a mixture of a solvent and a solute. In this beaker is a mixture of sodium chloride, table salt, and distilled water. The distilled water is the solvent. The sodium chloride is the solute. It's what we're dissolving. The same thing applies in our bodies. In the interstitial fluid, we have sodium chloride, we have water, we have potassium chloride, we have sugars, we have proteins. Those are all solutes in the solvent. Yes?
What slide did you put it on this?
Oh, it's not showing up?
It's like a whole.
Oh, swell.
In the cells first.
Oh, well, I think probably I must have cut that out. I must have rearranged it. Hang on a second. Yeah, do you see that? Membrane transport?
It's right under cell division.
Okay, are you on the, do you have the right slide?
Yeah.
Thank you. So,
Yeah, it's on there.
okay, thank you. I feel better now. Not that I would never make a mistake, so I will always own up to it. Okay. Now, so we deal with solutions and solvents all the time. Yeah. If you drink a Coke, for example, you're dealing with solutions. What's in the Coke? Well, you've got flavoring and you've got sugars and you've got a lot of acid in there. One of the reasons why soft drinks have a lot of carbonic acid in there, partly because of the carbonation to make it fizz, but also it takes a lot of acid to dissolve the corn syrup that they use for the sweetener now. So it's also why diet sodas have a great deal more of phosphoric acid and carbonic acid, because it takes a lot of, it takes a very low pH, a very strong acid to dissolve all the artificial sweeteners in the Diet Coke. Just so I throw that out there for you. So anyway, cell membranes have a problem in getting stuff into and out of the cell. Cell membranes are selectively permeable. They don't let everything through. Cell membranes are made of fat. So waters don't go, water and oil don't mix. If you ever had a greasy dish or something and you pour water on it, the oil is going to come to the top. If you ever mixed up Italian dressing, the kind you shake up in the bottle, and if it settles out, the oil goes to the top and the water and the vinegar at the bottom, right? Everybody's seen that. So you have to shake it up every time you want to use it, but they don't mix. The oils always separate out from the waters. Our cells are like that. They don't let water come across. For water to get into the cell, it has to have little tiny tubes for water to stick through, little tiny water channels. It's the only way they can get in. They can't get in across the membrane. Our cell membranes are very, very picky. They protect us, but in doing so, they keep out things like water. Salt has to have a special channel just for salt. Potassium has to have a special channel. So the cell membranes are selectively permeable, very, very restrictive as to what comes in and what comes out. Passive transport. Even if it's selective, we have passive transport, as I was mentioning earlier. If I were to take a dye pellet and throw it into a beaker, you would see a concentration of dye in one area of the beaker, but given enough time, the dye would circulate in the water in the beaker until it was uniformly distributed here. So, like the air in our room. The air in our classroom is uniformly distributed. There's no one area where we have more oxygen and less nitrogen than another. Like, oh, I can breathe better over here. There's more oxygen over here than there was right over here. It's uniformly distributed. It's in equilibrium. That's the term. Our cells want always to be in equilibrium with their contents. Water and salt, they want to be in equilibrium, and they work very hard passively to reach that equilibrium. Okay. The term diffusion, as I said, is a passive process where you go from a high concentration to a low concentration with no energy expended. It's all random, but ultimately we reach equilibrium. Now, why am I going on about this again and again and again? Well, let's get here to talk about types of diffusion. Simple diffusion, high concentration to a low concentration. Some things will simply diffuse into a cell. It's very easy. Some things will always go across the cell membrane from a high concentration to a low concentration. Anything that is soluble in fats or oils will slide across a membrane. Anything that's insoluble in fat. fat soluble substances. It will slide across. Oils can slide across the cell membrane. Hormones, chemical messengers that are soluble, that are made from fats, can easily slide across the membrane. Anything that is made out of proteins or with a lot of water or carbohydrates doesn't get through unless it has some help. And so simple diffusion tries to keep a lot of things out. Cells are real picky about what they let in and what they let out. So now this is the term I've been looking forward to bring up here. Osmosis. See, this is how cells work. There's a term called osmosis. Osmosis is a type of diffusion. In osmosis, osmosis is the movement of water across a cell membrane. It's movement of water across a cell membrane from high water to low water. So, osmosis. Movement of water from high water to low water across a membrane. Now, what does that mean? What in the world is high water and low water? Well, if you consider that water is found in solution in our bodies, we can measure the concentration of that solution. For example, our human body is made up is about 70% salt water. It's 0.9% sodium chloride. Now, what does that mean? Well, 0.9% sodium chloride find some space here. Here we go. Do it over here. 0.9% sodium chloride is, you would to make 0.9% sodium chloride, you would take a liter of water and you would take 9 grams of table salt to get to 0.9%. So you would have 0.9% sodium chloride is 99.1% water. If you look at it from a 100% basis, if you know that the concentration of the salt is 0.9%, then the amount of water in that solution is 99.1%. That's just how you mix these things up. If we have a 10% salt solution, if I have a solution of 10% sodium chloride, which is sitting right here, 10% salt, which we made up ahead of time. If it's 10% salt, how much water is it? 90%. It was on the board too, by the way. So right there. So 10%, whatever the solute concentration is, if it's 10%, if it's 40%, if it's 5%, if it's 0.9%, everything else is the water concentration. So if you have a 10% salt solution, it's 90% water and it's 10% salt. If you have 100% water, is anything, you can't get any higher than that, right? If you have distilled water, it's 100% water. So if you have 100% water, that's the highest you can get, and you have 90% water, which side is the highest, which is the highest water concentration, 100 or 90? 100, thank you. It's, it sounds like a trick question, but it's not. Osmosis is the movement of water from high water to low water. It doesn't matter if it's going from 100% to 90%. It's still the higher water to the lower water. So if you have a cell, and the concentration of water is higher outside than it is inside, then water will move into the cell. For example, if we have a cell at 0.9% sodium chloride, and we place that cell in 100% distilled water, it doesn't matter that this is, a human cell at 0.9% sodium chloride is 99.1% water. It's almost pure water, right? It's almost there. If you place that in 100% distilled water, the 100% distilled water still has the higher water concentration, and water will go into the cell. If you take a red blood cell and drop it into distilled water, the red blood cell is going to swell up. Because a red blood cell contains salt water at 0.9%. If you put it into 100% distilled water, what's going to happen is that cell is going to pop very quickly. It's going to, we call it lysing, or hemolysis, of rupturing of blood cells, because the water comes in so fast, because the water concentration outside the cell is 100%. Water concentration inside the cell is 99.1%, just enough of a difference for water to come in, and the cell pops. So that's why we don't give IVs with distilled water. So we have we have to consider the concentration of various solutions. So if we have a cell that is 0.9% and we place it into a solution of 0.9% sodium chloride, is anything going to happen? No, because the water concentration is exactly the same. If you have a cell at 0.9% sodium chloride, it's 99.1% water. Put that into a beaker that's 0.9% sodium chloride and 99.1% water. Absolutely nothing's going to happen. The water level is the same. And that's what we like to see in our bodies. The salt concentration in our plasma is 0.9%. The salt concentration in our interstitial fluid is 0.9%. Salt concentration in our lymphatic fluid is 0.9%. When it comes back to the plasma, it's still 0.9%. We don't like change at all. So, now, if there is a change, then water is going to move one way or the other. Person that gets dehydrated loses water. So think about someone who has been vomiting extensively or has diarrhea or both. Oh, by the way, speaking of that, did you read about the person in Italy a few weeks ago? He had monkeypox, COVID, and AIDS all at the same time. So, yeah, he was an overachiever of some sort, I guess. I don't know. But yeah, it's true. He recovered from the monkeypox, he recovered from the COVID, and he went into treatment for HIV. So he's recovered from all three of them, but he had all three at the same time. So anyway, I digress. So I just want to, I don't know why it popped into my head. I just had to talk about that. So a person who, oh, I know why, because he became severely dehydrated from nausea and vomiting from all three things happening at the same time. If you become dehydrated, you lose water. You lose water from your plasma. You lose water from your interstitial fluid. Because where does the water in your plasma come from? Anybody guess? Stuff we eat and drink. You know, you're taking a sip right now of your Gatorade, and that's going to, you know where that's going to go? It's going to go right into your plasma. That's what it does. You know, our bodies are designed that way. So everything we drink ends up in our plasma. Everything we eat ends up in our plasma. So that's where the water comes from. If we are dehydrated, if we are losing water from vomiting and diarrhea, we're losing plasma, we're losing interstitial fluid. So when we become dehydrated, the water concentration inside the cells and outside the cells don't match anymore. So when we become dehydrated, water tends to leave our cells and go out and try and reach equilibrium with the interstitial fluid and the plasma. So because when we lose water, our salt concentration goes up. We don't lose the salt, we lose the water. And this is where we get in trouble with dehydration, because It isn't, we lose the water. That's the first part. Then the cells respond by dumping water out of from their inside. The water inside the cells, they start releasing that to try and bring the interstitial fluid plasma back up to normal. And when cells start losing water, that's when bad things start happening. They start breaking down, and we start having, problems, neurological problems, digestive problems, and then we're dead in 3 days. So it only takes 3 days to die from a lack of water. So, and it's simply because the cells are doing what a cell does. Water drops, let's go out and reach equilibrium. And if we never bring that outside water back up. Conversely, if we overhydrate, that was underhydrating. If we overhydrate, what happens? You let's say that you start drinking lots of water. You have a whole case of Aquafina, your favorite bottled water. Okay, maybe it isn't. I don't care. You start drinking that one after another, because you think if some water is good, more water is better, right? I mean, if some is good, more is better. And what do they say? 8 glasses of water a day. We have to have 8 glasses of water, but you never notice that they never define the size of the glass of water. Most of us don't have a water problem. So you drink a lot of water. Where's that water going to go? In your plasma, right? Remember, everything we eat and drink ends up in our plasma and ends up in our blood. And now your water level in your plasma is rising. She's had like 6 liters of water. Overachiever. So you drink 6 liters of water, it all ends up in your plasma. You're now diluting your salt. You have your water concentration in the plasma is higher than it was before. It's now higher than the cells. Guess what the water does? It goes into the cells, and the cells start swelling up, and they may pop. That's exciting, particularly in the brain. So, too much water, too little water. Both of them are very bad. There was a really tragic example of this about 20 years ago in Seattle. There was I think it was Seattle. There was a contest. It was about the time that Nintendo brought out their original Wii. Anybody remember the Nintendo Wii? Okay, well, there was this radio station. I think it was Seattle, but it may have somewhere on the West Coast. Okay, and they had a contest. It was hold your Wii for a Wii, and they would have people drink water, and the one who could drink the most water and not have to go pee would win the Wii. It's a little play on words there, I guess. I don't know. Hold your Wii for a Wii. Well, no one thought through the physiological implications. I don't think radio stations do. Spend a lot of time on physiology, but anyway, so they had these people sitting there and they were drinking their water, and nobody was peeing. And they'd gone through a whole lot of water, and one of the contestants collapsed and she died because she had diluted her plasma and her interstitial fluid so much that the water was rushing into her cells. Her cells started swelling, her brain started to swell, and she started having ruptures and aneurysms in her brain, and she died because of that. So, and to be fair, nobody at the radio station knew that would happen. It seemed like a harmless, contest, but it wasn't. And she didn't even get the Wii either. So her family got the radio station, but ultimately, but she never got the Wii. So the point is, though, that nobody thought it through. Water levels in our bodies are very, very peculiar. I'll tell you, we refer to this as fluid and electrolytes in our bodies, and there is no faster way to kill your patient than to get their water levels and their salt levels messed up. That's just, once you get the water and salt levels out of balance in a patient's body, it's very hard to get it straightened out. So Our bodies don't like excessive amounts of water or to be dehydrated. So, okay, enough of that. The osmosis is, again, movement of water from a high water level to a low water level. Now, the counterpart from high water equals low solute. You know, if it's 99% water, the solute level is pretty low, like even in our sodium chloride, 0.9% sodium chloride, that's not a whole lot of solute. You know, you could say, and in some situations, this could be considered high water compared to something else. The point is that to know when we say high water to low water, high water always has very low solute, low water has very high solute. We've done different experiments in the past here with 40% glucose. 40% glucose. So we take 400 grams of glucose and mix it up with a liter of water. If it's 40% glucose, what's the water concentration? 60%. So now is that, so that would be, we would more than likely consider that a lower water concentration, because, you know, if we're comparing it to salt water at 99.1% compared to 60% water, yeah, this is the higher water, that's the lower water. So anyway, water will always move across a membrane. That's the key about a cell. Will always move across the membrane from high water to low water. And you're going to hear more about this in the lecture, but this is sort of one of those fundamental things about how movement across cells take place. Osmosis, high water to low water. Now, I've got an experiment running up here. This is a beaker of distilled water right here. I don't think you'd have a module mind. And this is a beaker of 10% salt solution. You can tell it's salt because it's cloudy. Got a lot of salt in there. So this is 100% water. This is 90% water. Within the beakers are, each beaker has an artificial cell. Now the artificial cell is made from what we call dialysis tubing, just like you use in dialysis equipment. This stuff is very thin. It also has lots of little tiny holes in there you can't see. So we can create an artificial cell. Now in the beaker with distilled water, there's an artificial cell containing 10% sodium chloride. So I filled up the cell, pinched it off, and put it in the beaker of distilled water. What's in the cell is 10% sodium chloride. Now, which side of the membrane has the higher water? What's in the beaker or what's in the cell? Or the beaker is 100% water, the cell is 90% water. The beaker has the higher water. Following what osmosis says, water should go into the cell. High water to lower water. 10% solute is 90% water. So 90% is less than 100%, so water should go in. How do we know? Well, I weighed the artificial cell before we started. The weight of the cell, the weight of the cell with the salt water was 30.39 grams. In the salt water, the 10% salt or 90% water, I put an artificial cell that was 100% distilled water. Which way will the water go there? Outside the cell. The cell is 100% and the water around it is 90% water. It's 10% salt. I weighed that artificial cell and it weighed out at 28.6 grams. Somewhere around 2 o'clock or 2:15, I want to weigh them again. And we're going to see how will we know if it worked, because we're going to see a weight gain or a weight loss. The prediction is that the water, the cell with the salt water inside of it in distilled water, water should come into the cell and we should see a rise in its weight, because water goes from high water to low water, 100% versus 90%. Here, water should go from high water to low water. The high water is inside the cell at 100%. The salt water in the beaker is 90%. This cell should shrink. It should lose weight. And if I did it right and didn't get things mixed up, we should see some changes here in these numbers. So we're going to let that cook for a while and go on there. So what do you need to remember about all this stuff I've been rambling on about? Diffusion is passive, from high concentration to low concentration. Osmosis is movement from high water to low water. You know, it took me 35 minutes to say all that. Okay, now, diffusion doesn't have to be in air or in a liquid. Here we see it's in a gel. In the past, we've taken crystals of methylene blue and potassium permanganate and dropped them into nutrient agar we use in microbiology. And the molecules of the potassium permanganate and the methylene blue actually start diffusing through the gel. And ultimately what will happen is they will come to, you can see who is diffusing faster. Smaller molecules diffuse faster than bigger molecules. Potassium permanganate is a smaller molecule than the methylene blue. And so it's spreading through the nutrient agar faster. And this is the experiment. And I've got the triple beam balances down there in the end, and we'll just wait and see. So, and you know, in the past we've tied this stuff off, but it works. You know, there's the example right there. The dialysis bag with 100% water in a beaker of 10% salt solution. That's what's happening there. And the one that's cloudy because of all the salt. And the other one is is the opposite. It is 10% salt in a dialysis bag of 100% water. So we'll see the results here shortly. Okay, let's move on. Any questions on all that? I think we'll see some decent results here, I hope. Let me go back and let's go into the other three tissue types. So can everybody that's zooming see the slide? It says human tissue types, muscle, connective tissue, and nervous. If you can't, let me know. You can't see it. Okay, let me try again then. I told me now. So, nope. Okay. Let me stop the sharing and try this again. Come on. There we go. Share screen. Okay, can you see it now? Great. Okay, thank you. Okay. So last week, we took a look at the epithelial cells. Oops. We took a look at the epithelial cells and we looked at the different types of epithelial cells. That is one of the 4 categories of tissues in our bodies. They're the covering tissues. The other 3 types of tissues are the muscles, the connective tissue, and the nerves. Now, muscles come in 3 categories. Skeletal muscle, which is also called striated. This is our voluntary control muscles. These are the muscles in our arms and our legs. You know, it lets us speak and talk, and I guess it's the same thing. Speak and swallow and move. We can manipulate our environment. These are the muscles of our arms, our legs, our hands, and our feet. They do what we tell them to do. They have a very distinctive pattern. They have stripes on them. We say that they are striated. Cardiac muscle is involuntary. We have no control over our heart. The heart controls itself. The heart has one characteristic similar to skeletal muscle in that it is also striated. Cardiac muscle, of course, is found only in the heart. And then the third category is smooth muscle. Smooth muscle doesn't look like any of the other two types. Smooth muscle is also involuntary. It surrounds all of our blood vessels. It's what we use to push blood around in our bodies to make sure that we have blood where we need it. And it can restrict blood flow. It can enhance blood flow. It also surrounds our digestive system and pushes food from the time we swallow it to the time it comes out at the anus. It pushes food through our entire digestive system through a series of muscle contractions. Okay, this is striated skeletal muscle. And these are the striations, the light and dark bands that make it look like it's striped. So we use, this is the shape of, this is very characteristic of skeletal muscle, of striated muscle. We call it skeletal muscle, voluntary muscle, striated muscle. This is the muscle that we can control. A couple characteristics here. These dark spots are cell nuclei. A lot of adipose tissue for energy. And then we have multiple nuclei. You can see there's a half a dozen nuclei lined up in a row here. When we were laying down muscle cells in utero, we were taking little cells and combining them into big cells. So we never lost the nucleus. It doesn't matter because we never replicate or copy, bless you, we never copy our muscle cells. Skeletal muscle cells don't reproduce. What we got when we were born is all we're ever going to get. Now, they can get bigger or they can shrink, but we're not going to grow new ones. We can, as we get older, as we go from childhood through puberty to adulthood, our muscles got bigger, the cells enlarged. If we work out and lift weights and do all sorts of stuff and eat lots of protein, our muscles will get even bigger. But we're not growing new cells, we're just enlarging those cells as they get stronger. So it doesn't matter if we have multiple nuclei. We're not gonna, the DNA in there isn't gonna be affected because we're not going to have, they're not gonna undergo cell division. So, skeletal muscle has very high energy demands. It's one of the reasons why you see the fat there. That's one of the reasons why there's lots of myoglobin there to carry oxygen for them. So we need oxygen for the cell to release energy stored in food. So characteristics, striations, lots of nuclei in here. Now this is smooth muscle, totally different than skeletal muscle. Smooth muscle has one nucleus per cell. It's also shaped like a spindle. We have no control over it. That was the other thing about skeletal muscle. We have complete control over our skeletal muscles. We have no control over smooth muscle. Our food is going to get pushed through our digestive system, whether we want it to or not. Smooth muscle is going to do it on its own. Same around our blood vessels. All of our blood vessels are surrounded by smooth muscle. The problem is that we don't have enough blood to fill up every blood vessel in our body. So we're always moving blood around to where we need it. On a hot day, we'll send lots of blood to the skin to radiate body heat so we can stay cool. On a cold day, we'll restrict the flow to the skin so we can stay warmer. And it's the smooth muscle that's going to close blood vessels or open them up for us, and we don't have to think about it. So each one of these nuclei represents an individual smooth muscle cell. Again, smooth muscle right there. Don't get this confused with the stratified squamous cells. They look similar in the slide, but each one of these little pink lines is an individual cell. Each nucleus represents a cell in here. And again, now, where do we find it? Where do we find smooth muscle? Well, here is a cross section of the small intestine. We have multiple layers surrounding the small intestine. Our smooth muscle there runs in two directions. We run lengthwise and we run in a circular pattern. So we can squeeze the small intestine in two different ways to push food through here. It's all controlled through the central nervous system. We don't have to think about this process. There is smooth muscle this is in the small intestine. Now in the heart is completely different from any of the other ones. Cardiac muscle cells only show up in the heart. They are striated, so they look somewhat like skeletal muscle, but they only have one nucleus per cell. The cells connect to each other and branch apart. They connect through what we call branching. You can see here are the striations here. You can see the striations in here. These little dark lines are called an intercalated disc. And whenever you have this disc, the muscle cells are going to branch to spread out to other cells. There's something about the heart completely different, is that when the heart contracts, all the cells in the heart contract together at exactly the same time. The two upper chambers contract, the two lower chambers contract at exactly the same time. Every muscle cell in the heart contracts with the same strength every time. There's no ripple effect across the heart. So so we don't have it. Some sort of selective control. In our arm, for example, we control the strength of the contractions by how many cells we're going to use, depending on what we're doing. If I'm moving this chair, am I using a lot of muscle cells for that, or am I using a few? Well, it depends. Depends on how heavy the chair is. This thing weighs several hundred pounds, and if I were to try and lift the tabletop up with one hand, I probably wouldn't be able to do it because it's very heavy. It's 300 or 400 pounds at least. So it took 4 of us to get it upstairs. So it is heavy. The point is, I would use a lot more muscle cells to try and move this around than I would to move the chair. I'm even using less muscle cells to pick this up. We control skeletal muscles. We determine how much force we're going to apply. If you're shaking hands with somebody, are you going to try and crush their hand, or are you going to try and be gentle?
Sure.
Yeah. Well, I guess it depends, doesn't it? Yeah. So it it's all variable. The heart has no variability. The heart will contract every cell, and that's the other thing. In skeletal muscle, we don't use every cell all the time. We don't use a lot of cells. We only bring cells into play when we need them. But in the heart, we use every cell every time. Every cell contracts in the heart to its fullest extent every time. So that's a big difference here. And the heart also is what we say autorhythmic. It starts itself. It doesn't depend on the nervous system. It's not under any outside control. So the structures of the heart, intercalated discs dividing up, the branchings, it's very distinctive. The only thing that's similar with skeletal muscle is that it is also striated. That's all. Here we see cardiac muscle cells, one nucleus per cell. You can see the intercalated disc, these dark lines here, and you can see how it branches apart. When you look at it there, yes, there are striations. There's an intercalated disc, and there's where it branches. So very distinctive, and only showing up in one place. Lots of cells, lots of nuclei, but not multiple nuclei. So one nucleus per cell. Okay, those are the three types of muscles: skeletal muscle, involuntary muscle, or smooth muscle, and cardiac muscle. Now, this category called connective tissue is sort of a catch-all. To be in connective tissue, you have to have three characteristics. You have to have living cells inside some sort of matrix, which can be a solid, a liquid, or a gel, with fibers. So this covers things like bone, because bone has living cells inside a solid matrix with fibers wrapped around the bone to give it strength. Or it can be blood. Blood has living cells in a fluid with fibers in solution that only come out when, like for example, for clotting. The fibers only form when we need them. So blood falls into the category of connective tissue. But the definition of connective tissue is a structure with living cells in a matrix of a solid or a liquid or a gel. So it can be something as like cartilage. Cartilage is connective tissue. You have cartilage cells inside the gel. You know, the soft, squishy gel of cartilage, you have living cells inside there, and then you have fibers reinforcing all that. So again, you go from, blood to bone to, cartilage, for example. So this is what connective tissue is. Again, it's sort of a catch-all in here. Got to have fibers, got to have cells, got to have some sort of matrix. So the fibers come in three categories. Collagen fibers. The most abundant protein in our body is collagen. Collagen is a very powerful protein. Collagen has an individual strand of collagen is stronger than a corresponding strand of stainless steel. Collagen fibers give our bones their strength. Collagen fibers make up the tendons that attach muscle to bone. The tendons themselves are so strong that they will snap bone before the collagen fibers themselves break. So it's very, very powerful. So collagen fibers, they work so well because they line themselves up in parallel. So they form these large sheets of fibers that run in the same direction. So it's like you would have, instead of having a thread to pull on something, you would have a million threads all pulling in the same direction with the same strength. So that's what collagen fibers do. Collagen fibers, elastic fibers. They are made up of the protein elastin, clever, and they're stretchable. Anywhere we want to have elasticity, like our skin, we're going to have elastic fibers. And then we have reticular fibers. Reticular fibers are the other, this is sort of, again, a catch-all. These types of fibers form the nets that hold our organs in place. You ever wonder why our abdominal organs fit into the abdominal cavity the way they do? It is these reticular fibers that form this, it's almost like a net that holds it together in place. All the organs are nested one on top of the other. And instead of all being piled together at the bottom of the abdominal cavity, they're held in place by reticular fibers. Okay. Collagen. Collagen fibers run in parallel. They have great strength. They're very, as I said, tendons and ligaments are made of collagen fibers. We wrap collagen fibers around our bones, so our bones will bend but not break. They'll have great strength. Elastic fibers, we find them in our skin, around the walls of our arteries, the large vessels that leave the heart. Arteries expand and contract as blood surges through them. If you've ever taken your pulse or ever had your pulse taken, your pulse and your heartbeat are two different things. Your pulse is nothing more than, in this case, the radial artery coming down along your arm. It's every time there's a surge of blood from the heart, the radial artery expands and then snaps back. And the pulse is the snapping back of the radial artery. And it can do that because of the elastic fibers. The arteries are elastic. As the surge goes through and it snaps back. It corresponds to the heartbeat, but they're two different things. We find our vocal cords that allow us to speak and make noise and sing and talk and at different levels and intensity are all elastic. Our airway has elastic bands around it so it can expand. Our bronchi have elastic bands around there. Our alveoli are elastic. So we have our ears are elastic. You know, we can do strange things to our ears and they snap back, just like our skin. It's all elastic. And then I said the reticular fibers are the mesh that holds things together. You know, some of our organs are nothing but reticular fibers. They're very squishy and they don't, it's hard to repair them when they're damaged. Okay, this is what we call loose connective tissue. Loose connective tissue. It has fibroblasts, which is nothing, you know, fibroblasts are what we get when we have scar tissue. Scar tissue is nothing but placeholders in our bodies. Scar tissue doesn't respond to stimulus. It doesn't grow hair. It just fills in a gap. There we have elastic fibers. There we have reticular fibers. And here we have nucleus, and over here we see collagen fibers. So loose connective tissue is sort of like the fill in the gap material. We find it like packing material. You know, it's sort of like the styrofoam peanuts in a box when you ship something that sort of fills in the gap for us. It's a mixture of all different types of fibers. We find a lot of loose connective tissue underneath our epithelial cells. They tend to form the basement membrane. So we sort of throw all these cells in there, these structures, so that the epithelial cells, like lining the small intestine, have something to hold on to. They become the basement membranes, for example. Again, where do we see it? Well, again, like I said, it is the, you know, a lot of times it forms the mesh with the reticular fibers to hold organs in place. Our spleen, for example, is surrounded by reticular fibers. Our spleen, which sits over here and helps to filter out old red cells, is a very squishy organ. You know, if you were to squeeze it, it would ooze out. You know, it's like sort of like squeezing Jello. You know, because our spleen is just not that strong. What it does is filter out dead, you know, red cells that have worn out. It recovers iron, and it tends to sequester the old hemoglobin for us, so we can use it to make bile. But the organ itself, first of all, it's not protected, and second of all, it's extremely squishy. It's sort of like the liver. The liver is pretty squishy too. So, you ever hear, you know, someone had an accident and they had a ruptured spleen? You know, blunt force trauma can just do horrible things to our spleen, and you know, it'll just, the force of it will tear it open. So, okay, now, dense connective tissue, the tendons and ligaments, lots and lots of collagen fibers. This is what collagen fibers look like in a tendon. They're woven side by side by side, so they have great strength. The tendons that hold our muscles to our bones are collagen fibers. And they are so strong that they will snap bone. You know, if they are stressed, if they are, you know, if stress is applied to a tendon, the tendon will snap the bone before the tendon itself tears. It's that strong. We call it dense connective tissue. So, and it's woven in a pattern where the individual strands, individual fibers reinforce themselves, much like in cloth, for example. So it has, as they say, great tensile strength. Each strand individually stronger than a corresponding strand of stainless steel. irregular connective tissue, dense, irregular, collagen and elastic fibers. We see this in our joints, like in our shoulder. Our shoulder has incredible flexibility to rotate around the socket. And it's held together by these collagen fibers and by elastic fibers. Not only can we move our shoulder and have it come back to its normal position, but we have great strength in our shoulder, thanks to the combination of elastic fibers and collagen fibers. Okay, cartilage. Cartilage is a type of connective tissue. It's made up of chondroblasts. Chondroblasts are cartilage producers. We have cartilage in our ear. We have cartilage in our nose. We have cartilage in our knees. We have cartilage in our trachea. You know, we have rings of cartilage in our trachea. And when we were still an embryo, we had a skeleton. We had a full skeleton in place 6 weeks after conception. So we're about the size of a little tiny tablet, you know, like maybe an Advil tablet or something like that. So we were about that big, and yet we had a complete skeleton in place made out of cartilage. And that was 6 weeks after the point of conception. And over the next 23 years, we gradually convert that skeleton of cartilage into bone. It takes that long. We don't complete the conversion to bone until we're about 23. At that point, we finally stopped growing. We finally stopped the conversion from cartilage to bone. It takes that long. The skeleton will get bigger and bigger, and while we're busy converting from cartilage to bone, we'll be growing and going through puberty and adolescence and young adulthood. And finally, about 23, the last of the areas of cartilage that are going to be converted are converted over to bone. But we retain cartilage in our tip of our nose, in our ears, in our airway. So we still have, don't forget the pads in our knees, the pads of cartilage in our knees that our femur rests on top of our tibia, the meniscus or menisci in our knees. It causes so much trouble in sports injuries, for example, because they wear out, they get damaged, they get torn. Those are pads of cartilage too. So we still have a few places left in our bodies that we've kept the cartilage. This is what cartilage looks like. This is hyaline cartilage. This is the stuff you find in the tip of your nose and in your airway and in the ears. Hyaline cartilage is laid down by cells known as chondroblast. A chondroblast I need some better markers. Anytime you see the word glass in there, it means it's a builder. Chondroblast is building cartilage. Osteoblast is building bone. So the chondroblast is laying down the gel. The chondroblast is laying down the matrix for the hyaline cartilage, and eventually that gel is going to solidify it's like making up Jello. You mix up Jello and you stick it in the fridge, and eventually it hardens. However Jello does it. You know what I'm talking about. I mean, who hasn't eaten Jello, right? That's what the cartilage does. It solidifies. The chondroblasts get trapped inside the gel, and then they become living cartilage cells. And when we have a living cell, we call it a site, in this case, chondrocyte, hyaline cartilage cell. Now, cartilage is different than all other cells in our bodies because cartilage doesn't have a direct blood supply. Cartilage has to get everything through diffusion. Oh, there's that word again, diffusion. Cartilage has to get everything through diffusion from the blood supply nearby, which is why cartilage injuries take so long to heal up, because they don't have direct nutrients being delivered to them. They don't have direct oxygen being delivered to them. It all has to diffuse through the matrix to get to them. So when they get hurt, healing takes a while. So only living cells in our bodies like that. So once they get trapped, once their gel forms around them, they're called a chondrocyte, and those little bubbles they're trapped in are called lacunae. And again, the only way they can get nutrients is for diffusion through that gel. Now, related to this is bone. Bone is also a connective tissue. It has living bone cells, the osteocytes, inside the hard bone. But they're a little different than cartilage. Bone has canals that has blood vessels and nerves pass through, very tiny blood vessels and nerves pass through, so that bone cells can communicate and be fed from each other. So this is what compact bone looks like. Now, we use osteoblasts that are, we call them the bone builders. Osteoblast has that same blast ending here. It lays down the bone matrix, and the bone matrix hardens around the osteoblast, and then those bone cells become osteocytes, and they're living cells, and they're contained within their own lacunae, just like the lacunae that the cartilage had, but now we have the osteocyte. But notice all the dark lines. Those lines are canals that connect not only from left and right and front and back, but up and down, because bone runs lengthwise. So cells communicate not only side to side, but up and down. And so we have blood vessels and nerves that run through these tiny canals. So every cell is in contact with every other cell, and our central nervous system knows what's going on in our bones. So they communicate this way, and they are fed this way. So we have living cells. It's easy to forget that bone is living tissue. You look at our skeleton there, and that's not real. But we look at it and say, well, you just see the bones. You know, we don't think about the fact that in our bodies, we have living cells all the time. Okay. Adipose tissue. Adipose tissue gets a bad, it gets a bad rep. It's not really, adipose tissue is not evil. Adipose tissue is just doing its job. Adipose tissue is a storage facility for fat so that we have energy. That's what it does. Adipose tissue will shrink and expand depending on the amount of fat stored in there. We have to have a certain amount of fat in our diet. We have to have fat in our bodies. We use fat for insulation. We use fat for energy. There is more energy in fat than there is in carbohydrates. Our body uses carbohydrates for energy, but there's only 4 calories of energy per gram of carbohydrates. There's 10 calories of energy per gram of fat. And yet our bodies are based off of carbohydrates. So anyway, what does this look like? Well, these are adipose cells. Adipose cells can shrink and expand. They are connective tissue because their fibers are elastic. And we have living cells. If you look carefully, you can see there's a nucleus right there. These cells will expand and contract as needed. The fat will be converted to glucose. There's a process known as gluconeogenesis, the creation of, it is the creation of new glucose. Neo means new, genesis means creation. So gluconeogenesis, the creation of new glucose. We see this happen a lot during pregnancy when the pregnant female needs to have glucose for herself in addition to the glucose that she's sending over to the baby. So, okay. And blood. Blood is a connective tissue. Living cells, red cells, white cells, floating in a matrix. The matrix is plasma, is salt water. The fibers are in solution. The fibers are used to form clots when we need it. But most of the time, the fibers are not activated. But it meets the definition of connective tissue. So I said connective tissue is sort of a catch-all. We go from liquid matrix to a solid matrix to collagen fibers and connective tissue and all sorts of stuff. So it sort of covers everything else. But blood is definitely a connective tissue. Meets the definition. And it looks like this. These are all these somewhat pink cells are red blood cells. The ones that are stained darkly with a large nucleus here, those are white blood cells. And these little tiny dots are platelets. They are cell fragments. Those are white blood cells with a large nucleus. And then everything else are the red blood cells. We have many, many more red cells per cubic milliliter than we do white cells. So typical blood count of an individual would be about 5 million red cells, 5,000 white cells, and about 500,000 platelets in the same size sample. So red cells carry oxygen. White cells are the infection fighters. Platelets are they form platelet plugs when we have a cut. They stop bleeding. Okay. Now the last category is the nervous system. This is a nerve cell. This is what we call a neuron. This large structure here, the large purple structure is what we call a neuron. And the neuron generally has structures on either end of it. We have axons and dendrites. An axon is how a neuron, a nerve cell, communicates with other neurons. It sends nerve signals, nerve impulses down the axon to a muscle, to a gland, or to another neuron. We also see, that's the neuron there, those little dark purple things are called glial cells or neuroglial cells. The little purple things, there's 6 different categories of glial cells, and they are the helper cells. There's a whole lot more glial cells than there are neurons. But the glial cells, as you'll learn in lecture, help to defend the neuron, help to feed the neuron, help to protect, defend, protect, destroy the neuron when it becomes damaged. The glial cells do all this for us. The glial cells help circulate cerebrospinal fluid around the brain and spinal cord. So they're very, very busy for us. They will also generate the insulation around the axon for us. And in some cells, they help to repair a damaged axon. There's the nucleus. Neurons don't regenerate. We don't make new neurons. We don't, whatever neurons we had at childbirth is what we have. We don't grow new ones. At least we don't think we grow new ones. So who knows? That may change in the next 10 years, but because there's been some stuff floating around that maybe we are growing new neurons. But generally, the conventional thinking is that whatever neuron, how many ever we had at birth is what we have. So, and there are nerve fibers, and usually these are the dendrites. A neuron uses dendrites to receive information and axons to send information. So, okay, and this. When I mentioned earlier about smooth muscle, this is a cross section of the small intestine. And here we see the outer covering of the small intestine. This is the serous membrane in the small intestine, which I was talking about in lecture the past 2 days. This is the outer layer, the outer covering of the small intestine. This would be the visceral membrane, as opposed to the parietal membrane. And underneath the serous membrane, we have 2 layers of smooth muscle. we have a longitudinal layer, and we have a circular layer. Longitudinal layer runs lengthwise, and the circular layer runs in a ring around it. So we can squeeze the small intestine in two distinct directions so that we can push food and digested food all the way through. And then this leads into, the outer layer is called the serous membrane because it's internal. Once we get a little towards the middle of the small intestine, we are exposed to the outside, and now we have the mucus layers. Submucosa, mucus, and then of course there's the lumen, the opening in the middle of the small intestine. Now, a couple more things and we'll wrap this up, and we will weigh the cells in the beaker. And now skin, we have some skin sections here on the slide. We have some models here too. Skins, this is where epithelial cells really come to play. The epidermis is mostly dead epithelial cells, mostly stratified squamous. And then underneath that, we hit the dermis where we have blood vessels. We don't have blood vessels till we get to the dermis. So we see the epidermal layer up here is stained in dark purple. And here we have the dermal layer. We see that hair follicles extend down deep into the dermis. They need it because they need the blood supply. And the hair shaft runs up through the follicle and extends out here. So here we have a model of just like the one you have right beside you there. This is a model of the skin. And we can see there's our epidermal layer. The outer layer of our skin is the epidermis, mostly dead until you get to the very bottom of the epidermis. The layer right above the dermis is where we have living skin cells, but everything above it is usually dead. Again, stratified squamous epithelial cells. There's the dermis. In the dermis, we have blood vessels. We have touch, pressure, and pain receptors. We have muscles. And then we get down to the third layer, we have what's known as the hypodermis, and that's where we find the adipose tissue in there. It's also where we see the sweat glands generating, generating their fluid that they're going to release. Oil glands we find up higher here in the dermis, but down here this is a sweat gland, this is a sweat gland, that's a sweat gland. These are oil glands here around the hair shafts. There's a receptor for pressure or touch. There's our hair follicle with our hair shaft in the follicle. There is our sebaceous gland that oils up our hair. It lubricates our skin, it lubricates our hair shaft. And if you think it doesn't do a good job getting our hair oily, don't wash your hair for a couple weeks to see what it's like, how unpleasant it might be. Because we do produce a lot of oil. We don't pay attention to what it does for our skin, but we definitely pay attention to what it does to our hair. But we lubricate our hair shaft so it doesn't get brittle, even though it gets really dirty when you don't wash it. But that's also because of the presence of the oil. There's the adipose tissue. Hairs are funny. There's no such thing as white hair. What we have is when a hair follicle, when the shaft loses its pigmentation, it becomes transparent. And so it appears to be gray. It appears to be gray. And if you have enough of them, it appears to be white because it's the light rays passing through the transparent shaft that makes it appear to be white. But there's no such thing as a gray hair. When we lose the pigmentation, it becomes transparent. So lots of people have gray hair. Older people, significantly older people have white hair. again, they've lost the pigmentation in there. It doesn't mean that the hair is wearing out. There are many people that have lost all their pigmentation. Their hair is white, and there's nothing wrong with that. The hairs are still alive. Hair cells are still alive. You just have no pigmentation. So, and it doesn't always have to do with aging either. There are people that start seeing their color loss very, very early in life, as early as their 20s. My dad started getting a streak of gray hair in the middle when he was in his 20s. Yeah.
My sister just recently, and she's like 12, but like parts of her hair is turning black.
Mm-hmm. So for whatever reason, she's losing the pigmentation in her hair. So a lot of it's genetic. My dad had it.
None of us in the family ever had that.
Genes are peculiar. Things can be hidden and then just suddenly manifest themselves. So I have 3 cousins on my dad's side that all have it. I dodged that bullet, but it's not unusual to have that occur. Now, 12 may be a little bit young, but I had a good friend in college that developed male pattern baldness starting at 18. So, and that's pretty unusual too. So, if you're all concerned, ask your doctor to look
Well,
at it.
they said like she had like a little checkup, but they said like they don't really know. Like it's just they don't think there's anything wrong with it.
Well, then it's probably just for whatever reason, her hair follicle cells have stopped making pigment on those, on those follicles. And that
Early sign of vitiligo.
could be, and nothing you can do, it happens. And some people keep their hair color their whole lives, and other people go gray very early or gray late. It just depends. So, who knows? Okay. Now, okay. The outer layer of our skin is called the stratum corneum. It is nothing but stratified squamous epithelial cells designed to wear off. It protects us. We have this clear layer is called the stratum lucidum. Again, dead cells. Stratum granulosum is below that. Stratum spinosum. And then when we get down to the stratum, the area right above the dermis, the stratum spinosum finally starts seeing live cells right as they get to the level of the dermis. The stratum basal or basement layer sort of mixes with the layer of the dermis. And so now you're seeing live cells. Also notice the fact that you have blood vessels in that region. So the cells can be fed. And there's a sweat gland, again, to help keep us cool when we get too warm. And we have pores released to the surface. Okay, and oil gland. The erector pili muscle it shows there, this is what allows us to, when we get cold and we get goosebumps, it's the erector pili muscle pulling on the hair shaft, trying to pull the hair shaft upright so it can trap more air on our skin, because air is an insulator. And the more hair we have on our skin, the more that stands up. We get goosebumps as we're trying to stand all of our hairs upright to warm us up. Doesn't do a very good job. We don't have enough body hair to do that for us, but that's what we do. And of course, there's the shaft. Now, one more thing to show you on this. Okay, another skin model. There we go. Now this one corresponds to this large model right here. And it's similar, but the important thing, I'll go ahead, if you feel the need to want to pass it around. Oh, so it's not that heavy, it's just awkward. Notice these bulges and bumps right here in the middle of this model. Right there, the thing's called the dermal papillae. You'll see them on that one on the far end. You know, you see a whole series of little bumps there. The dermal papillae are our fingerprints. The ridges on our fingers, they are characteristic of each of us that are, you know, a great identifier. Those little, at the far end of that model, you can see just like it is there, the dermal papillae, the loops and whorls are little ridges of tissue, and they're far enough underneath the epidermis and in the dermis that they don't disappear. And so our fingerprints are pretty much unique to all of us. Yeah, DNA is more unique, but the fingerprints are still unique. So that's where the fingerprints come from the dermal papillae pushing through underneath to give us those marks. So, okay, I think there we go. Now, let me weigh these two cells, and then we can call it a day. Okay. This is the 10% salt water, and this is a bag full of distilled water. This is 100% salt water, 100% water inside a beaker of salt water. Let's see if I got this dried off here. And the weight is 27.3. So the beaker, the artificial cell, went from 28.6 to 27.3. It lost 1.3 grams of water. Now you would expect that because you have 100% water in the cell, 90% water in the beaker. Water goes from high water to low water. It goes from the 100% side to the 90% side. So the cell gave up water and it did what it was supposed to do. Let's see what this one did. This is the salt water cell with the 100% water in the beaker. And its original weight was 30.39. Dirty. $37. It went to 37. Went from 30.39 to 37.4. It gained 7 grams of water. And you would expect that because the We had the 90% water, 10% salt inside the artificial cell. In a beaker of 100% water, water left the beaker and went into the cell enlarged. And you can even see it has gotten a lot puffier. I've seen these things turn and look like a hot dog, you know, like a little sausage or something. Anyway, that is everything I've got on this for today. Are there any questions? Okay, then we'll get out of here. I thank you for your patience.